Hello again and welcome to section 15.4 on RNA processing in eukaryotes. So our learning objectives here are to describe the different steps in RNA processing, understand the significance of exons, introns, and splicing for mRNAs, and explain how tRNAs and rRNAs are processed. So we just went through the process of transcription, and we have these eukaryotic pre-mRNAs that have to go through some processing steps before they can be translated. We'll also have those tRNA and rRNAs undergo processing so that they can help us with our very last step of translation, essentially protein synthesis. So when we look at that pre-mRNA strand, we are going to see that the genetic coding here, or protein coding, is not continuous. Instead, we are going to have these different areas that are called exons that are our coding sequences. I can't talk anymore today. I'm sorry. Sequences. And so these are going to be the important areas that are needed for gene expression. So we don't want to remove this area during the processing phase. We want to keep them. The other areas we have are called introns, and these are called intervening sequences. So you want to think that intron, I can't say it without the R, int part is for intervening, and this does not really code for anything, and therefore it's going to be removed in our processing phase. And you might think, well, this seems like a bunch of work that isn't very necessary, but the reason we have these introns is because they still appear to have some type of function that may regulate the genes and help with mRNA transport. So it's not that they're completely useless, it's just they don't have specific gene expression in here where we need a protein out of the material here. But once we do go through the removal process, which is called splicing, and we'll talk about that more in a couple slides here, that is going to be vital that we remove the introns properly, because we don't want any extra nucleotides hanging around. As we discussed in previous sections, this is going to change the coding for what amino acid is going to be attached to our polypeptide. So there are three important steps in pre-mRNA processing. We need to stabilize and add some signaling factors to the 5' end, and do the same at the 3' end, and of course remove those introns that we mentioned earlier. So let's talk a bit more about this, and this leads us into our 5' capping and 3' capping, which is called a poly A tail. So starting with that 5' end, we're going to add a cap with a molecule known as 7-methylguanosine that will help prevent mRNA from degradation and also helps our ribosomes recognize it so it can initiate translation. And after we go through elongation, the mRNA strand is going to be cleaved by an endonuclease between a specific area known as a AAU-AAA and a GU-rich sequence. And then we add this 3' end known as a poly A tail. Poly meaning many, so we're going to have approximately 200 adenines that are added to this 3' end by an enzyme known as poly A polymerase. And this helps to signal the transcript that it needs to travel to the cytoplasm so that we can go through the process of translation, and will also prevent degradation, just like the 5' end. So let's revisit that process of splicing that we talked about earlier. By allowing us to splice an mRNA strand, we are taking out those introns and essentially bringing the exons closer together. And this is done with a protein known as a spliceosome. So if you look at this mRNA strand over here, we can see that we've got 2 exons here with the intron in the center. The mRNA strand is going to kind of wrap around or within this spliceosome, and essentially it is going to cleave or cut the intron so that we get only the exon left. And this is a figure from your book depicting the same thing. You could see that within this spliceosome we also have those small RNAs, and we are going to excise the intron out and bring our 2 pieces of exons together. Now you may be wondering, how do we know exactly where to cut this intron? And it is because at the beginning and the end of each intron is going to be marked with a GU at the 5' end and an AG at the 3' end. And once we've gotten rid of all of these introns, we can have our what we call mature mRNA strand travel to the cytoplasm for translation. Now, we also said that within this processing phase, we also need to process our tRNAs and rRNAs. Our pre-rRNAs are going to be transcribed, processed, and assembled into ribosomes in the nucleolus. Remember, that is the structure that we find within the nucleus. And our mature rRNAs make up about 50% of each ribosome. With our pre-tRNAs, we transcribe them and process them in the nucleus and then release them into the cytoplasm because we are going to need to link a free amino acid to them. You can kind of see that pictured over here and in this figure from your book as well, where our tRNA is going to bind to its specific amino acid so that it's ready for protein synthesis. We do see that tRNAs have this 3D structure that is going to fold into position and attach that amino acid at the binding site, and then we have the anticodon on the opposite end. I think this is a little bit more of a simplistic illustration here, where we can see the amino acid on one end and what we call the anticodon on the opposite end. And what we mean by this anticodon is that when we are reading the mRNA strand, we had those triplet codons that we discussed earlier, right? And the anticodon is going to have that base pairing with the triplet codon. So if we have an A, we pair it with the U, we have a U, pair it with the A, and the G is going to be paired with the C. So if I continue to read this mRNA strand and the next 3 nucleotides read C, G, C, then the anticodon would be G, C, G.